Assalamualaikum warahmatullahi wabarakatuh. Let me introduce myself. My name is Nela Meriska Pratama. I'm from Biology Education major offering B. And now I will present my slide about the journal with the title Contamination of Microplastics in Pratama River, East Java, Indonesia, and its distribution in gills and digestive tracts of fish, Kombusia affinis. The first one, we are talking about journal identity. The writers of this journal are Nani Ratnapuwono, Yani Rizjani, and Agus Sugianto. And the year of this journal is 2021. The title of this journal is Contamination of Microplastics in Brantas River, East Java, Indonesia, and its distribution in gills and digestive tracts of fish Kombusia affinis. The name of this journal is Emerging Contaminants, and then volume 7, page 172 until 178. The second one is talk about background. Indonesia has many problems of aquatic pollution. Various studies have shown that microplastics have caused pollution of rivers to various degrees. Microplastics pose environmental issues related to the bioavailability of aquatic life. Microplastics damage the structure of several organs and tissues in fish. Respiration and digestion are the two primary routes of microplastic uptake. Ingestion was the main absorption pathway for microplastics among fish, which then collect in the intestinal tract. The third one is talk about research questions. Question number 1. Is there any microplastics were present in the waters and the gills and digestive tract of the Kombusia affinis fish? Question number 2. What is the differences in the abundance of several types of microplastics found in the various organ samples and locations? The fourth one is methodology. The first one, we talk about the study area in Brantas River Basin in East Java. Based on the ecological conditions of the Brantas River, 4 locations were selected, namely Batu City, Malang City, Mojokerto Regency, and Surabaya City. Batu represents the upstream portion of the Brantas River Basin, while Malang represents the central portion. Mojokerto Regency and Surabaya City are in the downstream part of the river. And then about the time, sampling was carried out from January to March 2020 with 3 replications per month. Microplastics are identified and measured in water and fish samples using from 2015 National Oceanic and Atmospheric Administration or NOAA standards. Kombusia fish were caught using nets with a mesh size of 0.2 cm. And then the type of microplastic was determined using a microscope with a 100-fold magnification Olympus CX23 LED lighting system, light emitting diode. And then in this study, data analysis conducted by using MANOVA to compare the abundance of different types of microplastics, gills, and the digestive tract of Kombusia affinis. MANOVA was used in 2 ways to assess the variation of microplastics types between the organs and among the organs of fish collected at the selected sampling sites. The fifth one, we talk about the result. The first result, as a result, 4 types of microplastics were identified in the water samples and the gills. This included fragments, fibers, films, and pellets. Microplastic of 4 different types, such as fragments, fibers, films, and pellets of less than 0.1 mm predominated in water, gill, and digestive tract samples. Their percentage values range from 76% to 100%. And then the second result is the average abundance value of microplastics during the 3 months of study on water samples showed an increasing trend from site 1 or upstream to site 12 or downstream. And then this is the picture of type of microplastics in water, gill, and digestive tract samples of Kombusia ovinis of the Brantas River. The first one is A1, this is a blue fragment, and then A2, this is a brown fragment, and then the B1, this is a red fiber, the B2, this is a blue fiber, C1 and C2, this is a transparent film, D1, this is a red pellet, and D2, this is a blue pellet. This is the diagram of the colors of microplastics found in the A, found in water samples, in the B, found in gill, and in the C, found in digestive tract samples. This is the diagram of the abundance of microplastics in water samples. Water samples revealed that the total microplastic concentration was lowest at site 1 and highest at site 12. As compared to the other three forms of microplastics, fibers was the most abundant, while pellets were the least abundant at site 1. And then in the site 12, the abundance of fragment is greater, and then the abundance of pellets was the lowest. So this is the diagram of the abundance of microplastics in gill samples. The least abundance of microplastics in gills was found at site 1, while it was most abundant was found in site 12. And then the greatest abundance of fragment was found in site 12. But also pellets were not found in the gills at site 1. So this is the diagram of the abundance of microplastics in digestive tract samples. The highest average abundance of microplastics in the digestive tract was at site 12, and then the lowest in site 1. The fragments were most abundant in the digestive tract, whereas the pellet was the least abundant. The sixth one, we talk about the discussion. Microplastics are generally described as plastic particles smaller than 5 mm with an indeterminate lower limit. Microplastics can accumulate in the gills and digestive tract through food. The color of the microplastics in the environment increases the consumption potential of Kombusia fish because of their similarity to natural prey. High population density gives rise to a vast volume of household waste that enters the aquatic environment and increases the microplastic in water. The abundance of microplastics in the digestive tract is significantly higher than in gills because the digestive tract is the dominant pathway for microplastic absorption. And now I will add some information about this discussion. The results show that the abundance of various types of microplastics is based on different samples and locations simultaneously. The combination of various locational factors with different sample types resulted in a significant difference in microplastic abundance values. The post-hoc test revealed that the abundance of each kind of microplastic differed from location to location. Furthermore, the concentration of each type of microplastic varies between organs, gills, and digestive tract. Fragments are the most abundant form of microplastics in water samples, gills, and the digestive tract. The great abundance of fragments is caused by the large of amount plastic waste, such as food packaging, bottles, or other plastic products that are discarded in the aquatic environment where it is degraded. And then the last one is conclusion. This study looked at the form, scale, and color of microplastics which present in water, gills, and digestive tract samples of Kombucha affinis. The greatest abundance among all types of microplastics is size of microplastics less than 0.1 millimeters and black color. Microplastic levels are higher in digestive tract than in gills. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.